Hello everyone, welcome back to our channel, and today we'll talk about Agarose Gel Electrophoresis. So electrophoresis, as you all know, is the separation of DNA or protein in the presence of an electric field. Now electrophoresis could be of two types: agarose gel electrophoresis and polyacrylamide gel electrophoresis. Now both of these two can be used for the separation of DNA, and the choice of the gel depends upon the molecular weight of the DNA we are trying to separate. So for agarose gel, it is normally used when we are trying to separate DNA molecules ranging from about 100 base pair to 25 kilo base pair. Now why does DNA migrate in the presence of an electric field? We know DNA is negatively charged owing to the presence of the phosphate group. So when an electric field is applied, DNA moves from the negatively charged electrode, which is the cathode, towards the positively charged electrode, that is the anode. Now there are different factors which regulate the rate of migration through this agarose gel. Now before going into that, let us know what is agarose. So agarose is obtained from seaweeds, Gracilaria and Gelidium. So Gelidium and Gracilaria are the commercial source of agarose. Basically, agarose is the polymer which is composed of alternating chains or alternating D or L galactose residue. Now these chains form helical fibers which further aggregate into supercoil structures. So you can imagine agarose gel to be a three-dimensional mesh with a varying pore size. And depending upon the concentration of agarose, the pore size of the gel matrix can range from 50 nanometer to about 200 nanometer. Okay. Now let's see what are the factors which affect the DNA migration through the gel. First and the most crucial is the molecular size of the DNA. Now it has been seen that the rate of migration is inversely proportional to the log 10 of the nucleotide pairs. So this is the log 10 of base pairs and this is the rate of migration, which means that larger DNA molecule face greater frictional drag and they are retarded to a greater extent when moving through the gel matrix, whereas smaller molecules migrate faster through the gel. Next is the concentration of agarose. So as I have mentioned previously, that the concentration of agarose determines the porosity of the gel matrix. Now it has been seen that if you use high concentration of agarose, so if you are using high concentration of agarose, say about 2 to 3% of agarose, the pore size of the mesh would be small. Pore size of the matrix would be small, which would hamper the movement of the larger molecules, whereas smaller DNA fragments will migrate faster. So alternately, if you're using a low concentration of agarose, for example, 0.5% or 0.8%, the pore size of the matrix would be large and larger DNA fragment would move easily through this sieve-like structure. Third is the conformation of the DNA. Now, DNA can exist in three forms. One is the supercoiled form, say, which is something like this. It is very compact and supercoiled. Second form is the nicked circular form. So it is circular, but there is a nick somewhere over here. Or linear DNA. Now you can understand from this diagrammatic representation that a linear DNA would face more frictional drag compared to a supercoiled DNA, which is more compact. So by this, the rate of migration of supercoiled DNA is greater than nicked circular DNA, and linear double-stranded DNA has the lowest rate of migration. And finally, the applied voltage. And it has been seen that at lower voltages, the rate of migration is directly proportional to the voltage applied. So if you increase the voltage, the rate of migration of the DNA increases, whereas if you lower the voltage, the speed reduces. So these four are the main factors which governs the migration of double-stranded DNA through an agarose gel matrix. Now, one of the most important components apart from the agarose gel is the electrophoresis buffer which you are using for DNA separation. Now, you cannot use water because water would tend to get heat up easily, leading to the melting of the gel. So you need to provide a buffer which would maintain the pH of the solution as well as provide the necessary ions for conductance. Now the two most commonly used buffer are TAE and TBE. TAE stands for tris acetate EDTA, whereas TBE stands for tris borate EDTA. Now, there are two factors which needs to be considered when choosing the buffer. One is the buffering capacity. What do you mean by buffering capacity? That is how long the buffer would be able to provide ions to support the migration of the DNA. And the other is the resolving power. So what is resolving power? Resolving power is how sharply the DNA of different molecular sizes could be separated in the gel. Now, it has been seen that between TAE and TBE, the buffering capacity of TBE is much higher than that of TAE. However, if you consider the rate of migration or the resolving power, TAE becomes the buffer of choice. And in case of TAE buffers, DNA has been seen to migrate almost 10 times faster compared to TBE. So to obtain greater resolution and sharpness of the band, TAE is chosen over TBE. So these two are the most commonly used electrophoretic buffer. Now DNA is invisible. You cannot see it by naked eye. So to ease the process of loading the DNA into the well, you need to mix the DNA sample with a gel loading buffer. So if this is an agarose gel, you have vials like this where you put into a DNA sample. So generally the DNA sample is mixed with glycerol, which increases the density of the sample and it allows the DNA to sink into the well. Another component of gel loading buffer is the dye. The two commonly used dye are bromophenol blue and xylene cyanol. Now these two dyes not only eases the process of DNA loading, where you can easily see that the sample is entering into the well, but it also helps in monitoring the migration. That is how much the sample has run into the matrix. And generally these dyes do not hamper the rate of migration. And it has been seen that bromophenol blue migrates with double-stranded DNA of size 300 base pair. So you can get an idea by looking at the dye front that how long your gel has run and where can be your desired band of interest. Now let's move to the final stage of agarose gel electrophoresis, which is the visualization of the DNA. Now for the process of visualization, we use a fluorescent dye, which is ethidium bromide. Apart from ethidium bromide, cyber gold could also be used, but ethidium bromide is the most commonly used dye for DNA visualization. Now, ethidium bromide intercalates within the DNA. So, it lies perpendicular with the DNA bases above and below it. Now, normally DNA absorbs in the UV range of about 254 nanometer, whereas ethidium bromide absorbs at 302 nanometer and 366 nanometer. Now, ETBR can bind to both single-stranded and double-stranded DNA. However, the fluorescent signal increases to about 20 to 30-fold upon binding to double-stranded DNA. So, when double-stranded DNA intercalated with ETBR is subjected to UV radiation, it emits light in the range of 590 nanometer, which is the orange-red region of the visible spectrum. So, in a nutshell, you cast a agarose gel, you load your sample, you run it in the presence of a suitable electrophoretic buffer. Upon the successful completion of the run, you put the DNA under UV light. So this is your gel. It is transilluminated with UV. It would absorb at 302 nanometer and emit in the orange-red region at 590 nanometer, showing the position of the DNA in your agarose gel. So hopefully you have understood the concept. Next time we will come back with another new biotech topic.